Good afternoon, everyone. Uh, I'm bringing you back to basic research. So today's topic, which I'm covering, is identifying appropriate technique for basic research studies. Now, this is a very, I would say, a weak link in study design for the researchers, which generally does not receive the adequate focus. And yesterday we had a discussion with Dr. Haldan where he was talking about the translation potential of the research. Generally, this is the stumbling block because when we are designing the study, we don't exactly focus on how the technique that we're using for the research is going to be taken up when we're doing the translation. This one. Okay. Better now? Okay. So I'll quickly take up some important points to keep in mind when you are looking at deciding the technique to perform your research. Most of this has already been discussed under different headings yesterday, but it is like a summary of the points to keep in mind when you are finalizing your research technique. So always in your mind, this thought should be there. The technique that I'm going to use, is it the right fit for the conclusion that I want to draw? And always keep in mind the translation potential, the result, how that result is going to be used. So will the technique still be useful? Will it still give the important information when we take it to the translational aspect? Some of the basic points which you have to keep in mind already discussed. You have to talk about the sensitivity, specificity, whether they are adequate, whether you have a better study with better technique which has more sensitivity. If you're talking about screening, if you're talking about the diagnostic approach, whether you have more specific tools to achieve the same result. So these are some basic considerations discussed multiple times yesterday. I'm not going to. Something which we generally miss. You are using a technique to give a result, but is that result getting affected due to cross-reactivity from other factors which you not consider? Are you including those factors which are causing the cross-reactivity in your observation to carry out the discussion and finally reach a true or correct conclusion? So this can be one stumbling block which you have to keep in mind. Is the technique that you're using or the result you are achieving, is it dependent on other factors? These examples. So are you considering factors also in your mind? Dependency can be a very, very important factor in, you can say, making your result non-conclusive or making your result doubtful, whether it is meaningful or not. So always keep in mind the factor that you're using or the technique that you're using, is it dependent on factors which you've not included in your study design? Very, very important. What are the limitations of the technique or the analytical process that you're using? Are you including that in your observations? Are you including that in your discussion? Are you including that in the conclusion? Because if the limitations are not discussed, then when you go to the translation aspect, the problem will be there. Just in the first session, Dr. Rucha was saying that they had standardized with 60 microliter, and when they went to the implementation, they were told anything having more than 30 microliter, they will not be implemented because it is not practical to do. So limitations become a very important factor when you are deciding the technique for the basic research. This ties up with the other factors that we have previously mentioned, whether the technique that you're using to reach to a conclusion in your basic research, is that technique enough? Or you need more information to reach the conclusion? For example, if you're talking about oxidative stress, and you say I'll measure the glutathione levels, is that enough? So adequacy is an important factor. Otherwise, you will find when you're submitting your article for submissions, the reply will be okay, your work is okay, but the parameters are not adequate to justify publication. So at that point, you'll find that good journals are not accepting your results, although the topic, the structure, the whole study which has been conducted is good, but the techniques that you've used are not adequate to reach the final conclusion. Adequacy is a very okay. Last, when you are choosing a technique, one important question comes to mind whether the technique will be available to you, whether it is accessible to you or not. So here, what you should keep in mind is depending on your sample size, will it be justified to create the infrastructure for the technique in your institute, or it will be more cost effective to outsource? And if you are thinking of outsourcing, whether the facilities are available to you to outsource or not. 5 years, 6 years back, the whole genome sequencing was not an available or accessible technique to us. But now, even institutes who do not have the NGS in their own infrastructure, in their own institute, can think of outsourcing, because a lot of institutes will be willing to tie up with you, or a lot of private players are available who will perform the NGS for a reasonable price for you. So this is an important consideration also when you're thinking of technique. And this ties up with the adequacy of the technique. Means if you're looking at a technique which is adequate, but you find that you're not able to do it in your own institute, or the expertise is not available to do it in your own institute, can you tie up with others? And if the answer is no, then you have to look at other techniques. So that is important consideration when you are deciding on a technique. So I'll talk about these factors. I'll take some examples to tie up all this information with you. I'll take examples at the planning stage. I'll take examples at the publication stage. I'll take also examples at the implementation stage when these factors come into play. For example, you know that the reference values that you use in the clinical setting, most of these reference values are not generated in our country. They are being used from the other places, mostly from the US. Just like we have our NFHs, they have their own data, which is NANEX, which is also a periodic exercise where they collect data from the population. So we are using lots of reference values coming from that data. One important data from that is hemoglobin levels, which is an important doubtful reference value for us. A lot of clinicians will always say that we are using the reference values from the Western population, which is not applicable to us. Our body structure is different, our BMI is lower, our genetic composition is less, our dietary habits are different. So we should have our own reference value for hemoglobin. So we are in the process of designing a study where you are going to use the hemoglobin as the value you are measuring in the general population. Now, when you are doing the data collection for the general population, you will identify people who are healthy, apparently healthy, and then take that data. And from that data, you will try to find out the ranges, the mid value, the average, the confidence intervals, like that will do. But what you have to keep in mind is a person who is slightly away from the apparent normal. Let's say the apparent normal is 12.5 gram per liter of hemoglobin, and a person who has a value of 12 gram per liter, that person will also appear to be normal. Or a person who has even 11.5 gram per liter will also appear to be normal. Now, this problem of a lower value giving an apparent normal presentation, how you can decipher whether it is normal value, whether it has to be included, are there some parameters based on which we can decide whether this is a normal value or it is due to some deficiency. So when we are planning the study to observe the hemoglobin in the normal population, we also have to keep in mind to include parameters which affect the hemoglobin. If you're not collecting the data for the iron stores in the body, if you are not collecting the data on the morphological picture, you will not be able to select the normal data and your result will be biased, which will be challenged later on. Did you see the morphological picture of the people you've included? You're saying, okay, the result of the study says no, for Indian population, the value should be 11.5, not 13. and you've not looked at morphological picture, large number of those individuals were having iron deficiency anemia, which could have been seen in the morphological picture, which you could have identified using the iron stores, but you've not done that. So in that case, the result will be just thrown out of the door because you don't have the supporting data to justify that your data is accurate. It is dependent on multiple factors which you've not seen. So your result is weak. It will not be accepted. So this is something which you have to look right at the planning stage. I'll give an example where the data can be confounding and the study design will give you confounding results, which might even go to publication. Last few years, we have a factor called ischemia modified albumin. It is counted as recently as an important factor in identification of myocardial infarction. Because infarction is there, it will cause modification to the albumin. And the beauty of this parameter is you can detect it within a few minutes. Because if ischemia is there, circulation albumin contains, and it will be modified, you can quickly identify it. So if you plan a study where you are measuring the ischemia modified albumin and you compare it with the current gold standard, which is troponin, cardiac troponin we are measuring. And if you plan the study in admitted patients where you can quickly find out the patient after the event has occurred, and you compare the changes in IMA, ischemia modified albumin, and cardiac troponin levels. So here, if you don't consider the factors for the different tests that you're using, there can be a fallacy in your report. One of the reports published said that IMA is even more sensitive and more important than cardiac troponin, where the case subjects were admitted patients. But before reaching to that conclusion, what you have to keep in mind, the cardiac troponins are not detectable earliest before half an hour if it is a massive MI, or 1.5 to 2 hours if it is a moderate or a mild microinfarction. If you're collecting samples less than half an hour after the event, then you are unlikely to find the cardiac troponins to be elevated. Whereas the ischemia modified albumin, it is coming out in 2 to 3 minutes. It will always be there in the samples. So based on that, if you derive the conclusion that ischemia modified albumin is more important, more suitable to identify the microinfarction than cardiac troponin, then your conclusion is flawed. So we have to consider the various factors which affect the result and its interpretation before we choose the best fit. So these are the factors which you have to keep in mind. I'll talk a little bit about selecting or identifying new biomarkers, where we will focus on some of the techniques and what are the different aspects of the different biomarkers and different techniques which can be used for doing this. Okay. So a little bit about identification of biomarkers. Very quick background of biomarkers for people in basic research. They will say, "Oh, such a pedantic thing, such a boring." You already know this, but I hope it is slightly helpful for those from the clinical background who lost touch from these points. Okay, so biomarkers, biological markers in medicine, they're used to detect a disease, discover drugs, and monitoring care of patients. When we are talking about basic research, we mostly focus on molecules or genetics or expression, but in clinics, it can also be imaging. I'll not be covering the imaging aspect here. I'll mostly focusing on the molecular aspect of the biomarkers. I'll not talk about imaging as a biomarker. Okay, so based on what you are trying to see with the biomarker, you can have the predisposition biomarkers, you can have diagnostic biomarkers, you can have prognostic biomarkers, and predictive biomarkers. So based on your study design, what you are trying to give as the conclusion, you have to choose the appropriate markers. So, and based on that, you'll choose the appropriate technique for the study. Some examples of biomarkers: they can be hormones, they can be receptors, they can be enzymes, They can be other molecules. So they can be carbohydrates, they can be the oncopetal genes, and molecular biomarkers based on their composition can be peptides, proteins, lipids, metabolites, even carbohydrates, nucleic acid. So all sorts of molecules can be biomarkers. Additionally, they can be distinguished as disease-causing markers and susceptibility markers. So, what molecules will be included in which headings? So, there can be distinction based on that also. Which ones will be indicating exposure? Which ones will be indicating susceptibility? Which ones will be indicating disease? So, like I said in the beginning, obvious point of biometric evaluation is sensitivity and specificity. These are the two very, very important, and you can say the baseline, minimum baseline, which you have to keep in mind when you're talking about biomarkers. But at the same time, authentic and standard methods of sample collection are very, very important when you're talking about biomarkers. I'll take the example of molecular epidemiology. In the past, epidemiology has focused on the causation from a lifestyle, from a diet, those sort of things. But now epidemiology is shifting towards the molecular aspect of epidemiology, which is the genetic factor which is causing the disease, which is the expression regulation which is causing the disease, which types of proteins are causing the disease, what are the different metabolites which are associated with the disease. So now this is one part where the public health experts and the clinicians also need to know a little bit about the biomarkers. When you're talking about epidemiology, then also you have to look at this aspect. So I'll quickly try to show the different techniques which can be used when you are analyzing the different aspects of the molecular epidemiology. For example, if you are looking at DNA, you are doing an exploratory study, you might want to look at the whole genome. So in that case, you need to have facilities for the next generation sequencing where you can look at whole genomes. If you already have some preliminary studies which give you some preliminary data, you can have a focused discussion on the DNA. You can talk about the polymorphisms, you can talk about the epigenetic changes. So the techniques will correspond to that. You're looking at polymorphism, okay, the polymerase chain reaction would be good enough. If you're looking at epigenetics, changes. So the modification in PCRs which focus on epigenetic changes, those could be the technique that you're looking to employ. If you are talking about the regulation of transcription, that is gene expression, which is affecting the epidemiology, then you want to look at the RNA. Here, one important aspect can be RNA profiling. It is similar to the whole genome sequencing. We are looking at the whole composition of the RNA from the body, and based on the samples, you are finding the potential markers. Otherwise, if you already have some existing data, you can look at microarray. You can look at microarray, or you can look at the specific transcription of the molecules. Most important, most commonly employed one is the protein, and for which we have the proteomics. And nowadays, we are also looking at the other components which will come under the heading of metabolomics. So these are some of the different aspects of the markers. Based on that, you will have the different techniques. I'll pick up proteomics and quickly flash some of the techniques which you can commonly use when you're looking at identifying a new biomarker. And go to proteomics. Proteins are preferred because they can be detected in very tiny amount of sample. If you choose your samples correctly, if you choose the target protein correctly, then generally they have good sensitivity and specificity. And that is why proteomics is one of the commonly used avenues for identifying the new biomarkers. How to use the proteomic approach to identify a new biomarker? It will have multiple steps. So you have to identify at which step your hypothesis is focusing, and based on that, you have to choose your techniques. If you already have some identified proteins which you want to either validate or you want to associate with a particular disease, then you would be entering this whole flowchart somewhere in between. For example, if you already have some preliminary data and you want to verify, so we are only looking at verification of proteins. But if you're looking at identification of a totally new marker, then you have to start from scratch. First, there has to be extraction and separation of the proteins, identification of the proteins, and then the verification of the proteins. Typically, when you're doing this identification of proteins from extraction, you're using the mass spectrometry. So in that case, you have to do a lot of database search, you have to do the protein-protein interaction studies, you have to do, finally, you have to do the statistical analysis. So again, what is your hypothesis that will decide which technique you are going to use from this whole gamut of the process? Okay, so I'll quickly run through the different techniques at different steps, and I'll not go into the details because we have time constraint. And just give an example that we have lots of options of choosing the specific technique based on the different factors that we discussed previously. For example, if you're talking about separation, you can base your technique on multiple factors. You can do the separation based on solubility. You can do the separation based on molecular size. You can do the separation based on molecular charge. You can do the separation based on affinity binding. Different techniques exist in each of the domains. So, for example, the next techniques will be size dependent. You have the dialysis. Okay, you have dialysis, gel filtration chromatography, ultracentrifuge. These are dependent on the size of the protein. Then you can have various procedures which are dependent on charge. Commonly, it is the chromatography and electrophorese. Different types of chromatography and electrophorese are there depending on the property of the protein. If it is a charged protein, you can go for ICMR chromatography, or you can use the high performance liquid chromatography. If it is charged particle, then you can go for electrophoresis, depending on whether you want to identify the protein or whether you don't know about the protein, but you're trying to separate. Different techniques will come into the picture. Okay, so we can have anionic chromatography, you can have paper chromatography, you can have polyacrylamide gel electrophoresis, you can have isoelectric focusing. And lastly, based on affinity binding, you can have more techniques: affinity chromatography, absorption chromatography, presentation of antibodies. Each of these parameters will have their own pros and cons. For example, if you're using the property of affinity, you tend to get very, very pure proteins. Where you want to do the analysis, the new protein is there, you don't know anything about it. So you go for affinity chromatography, so you get the pure protein. But if you already know a little bit about the protein, you want to do the identification. In that case, you can go for the separation based on charge, where you get reasonably pure proteins, although they're not very pure for doing the analytical or functional studies, but it will serve your purpose because you are wanting to do the identification. So based on your target, you can choose the suitable technique. Okay, so this is an example of affinity chromatography. You can also merge techniques. For example, you have two-dimensional electrophoresis, which is having two techniques. First, you do the isoelectric focusing, then you do the SD stage to get the more clarity. If you have a complex mixture, you can merge techniques to get the more clarity about the output. Okay. This identification of proteins relates to when you're doing the separation of proteins using the mass spectrometry. Various techniques can be there. And mass spectrometry is one of the ideal methods for identification of the proteins, which is currently used because it is able to identify a very large number of proteins in one given sample. The others are more, you can say, limited in number of the proteins that they can identify. And coming to verification of proteins, commonly used technique is ELISA. You can have the Western blot also. Sometimes when you're doing the identification of proteins using MS, you might not be having the antibody because it's a new protein. In that case, you have to go for either protein sequencing or you can do the peptide mapping if you use the mass spectrometry for the separation of the proteins. Lots of database. This is for the mass spectrometry data. Lots of databases are there which can be used for searching and identifying the proteins. Lots of scientists are already working. A lot of data is already there for the proteins, and it is quite likely that the protein you have separated or identified already exists in one of the databases. It will help you to quickly pinpoint which protein you're talking about. I'm not going into protein-protein interaction and statistical analysis. Statistical analysis has already been discussed. Lots of, again, softwares are there which can be used. Commonly, we use the SPSS or the GraphPad Prism at the institutional levels, which can be used for statistical analysis. Last 2 minutes, I'll just take an example of the factors which come into play at the level of implementation. So nowadays in the microbiology lab, where we talk about antimicrobial resistance, the classical, the gold standard is where you do the culture studies, and based on culture, the phenotypic output is there, and then you get the result of the antimicrobial resistance. But with the identification of the genes which are responsible for those, you can say, resistance, we have this genotype phenotype based AMR, where once the identification of the microbe is there, we quickly do the genotypic analysis. We have the panel of genes which are causing the resistance, and based on that, we can immediately tell the clinician, okay, this particular organism we have isolated, and it is, let's say, penicillin resistant, and you need to modify your treatment accordingly. This result comes in a few hours after identification, whereas the traditional phenotypic result will take 2 to 3 days for the culture to develop and the result to go out to the clinician. It can be very, very important in managing very critical patients where if MRSA is there and you're giving, let's say, ampicillin or penicillin, the patient would not respond. You need to escalate the treatment to higher drugs, maybe vancomycin or something like that. So there it becomes very, very important. This is called syndromic molecular diagnostic panel, and it is considered to be very, very, very useful because it talks about the mechanism of antimicrobial resistance. But if you don't understand the limitations of this technique, it can become a very, very, you can say, problematic result for managing the patient, which can result in the mismanagement of the patient. Quite a few times we see that the genotypic result does not match with the phenotypic result. It doesn't mean that you have done the genotypic analysis and you're not doing the phenotypic analysis. We still do the phenotypic analysis, which is going in the background. It will give the result after 2 to 3 days. But quite a few times we find that the genotypic result doesn't match the phenotypic result. Suppose you have identification of gram-positive cocci, and then you do the genotypic analysis where you find that, okay, it is staphylococcus, and you find that the MEK gene is not there or there. Based on that, you give the susceptibility result has been given that, okay, this is susceptible or it is resistant. Let's say MEK gene is not there, and we say that it is MRSA. But when we do the phenotypic analysis, we find that, okay, staph was there, but we have two. Staph aureus is also there and hemolyticus is also there. And the resistance was there, the MEK gene was absent in the hemolyticus and not in the staph aureus. So in that case, we would not have required to escalate the treatment to the beyond penicillin to maybe vancomycin. So this is another point where you have to keep in mind the limitation of the study even at the level of implementation. Not only during the statistical research, but at the level of implementation also, we have to keep in mind the limitations of the techniques that we are using. So I'll conclude here with the final considerations. You have to keep in mind the requirement of your hypothesis question when you're choosing the technique. Whether the expertise is available or not, or whether it is accessible to you or not. Similarly, infrastructure and self versus outsourced. Sensitivity and specificity are always the basic minimum that you have to keep in mind, and whether the results are valid, whether they are accurate, and whether they are precise. These have already been discussed previously. So this is all I wanted to bring to your notice. Thank you very much. Any questions?
Thank you, Dr. Chandra, for very crisp presentation and very informative. You have covered 4 or 5 very important aspects of research studies. Thank you for your. So, in the interest